Let's go ahead and begin. So in today's video, we're going to be talking about B cells. This is a very important video because B cells play a crucial part in your immune system. By the end of this video, you will completely understand what B cells are, B cell maturation and selection, B cell function, the antibodies they produce, their function, and also the different types, clinical relevance, and also at the end, we'll throw in a few USMLE practice questions, not only to test your knowledge, but more importantly, solidify it for the long term. Be sure to check out the healingpowerofnature.com and also our Patreon for some of the best materials on the market. If you're struggling with this information, I want you to imagine your body as a fortress. It's constantly under attack by invaders like bacteria and viruses, but deep within this fortress, there is a secret research lab run by elite scientists. These elite scientists are your B cells. What is their mission? To analyze threats, create targeted weapons like antibodies, and build memory database. The memory database helps to stop future invasions before they even begin. So B cells are produced in the bone marrow. Before leaving the bone marrow, B cells must be screened for autoimmunity. So first what happens is a process known as negative selection or central tolerance. Immature B cells that bind too strongly to self-antigens are either deleted or edited, known as receptor binding. This prevents autoimmune diseases like rheumatoid arthritis or lupus. Then there's survival signals. B cells that do not strongly recognize self survive and enter circulation. So if there's defects in negative selection, autoimmune diseases will arise. Why? Because the B cells will attack your own tissue. They start as naive workers or naive B cells, but when trained or activated, they begin producing highly specialized products known as antibodies tailored to fight specific invaders. Once B cells are activated, they function as the primary producers of antibodies, which neutralize pathogens, enhance phagocytosis, and activate the complement system. Like I stated, they start as naive workers, but need to be activated. They begin producing highly specialized products or antibodies once they are activated. Antibodies are tailored to fight specific invaders. So, plasma cells, these are the antibody secreting machines that arise from activated B cells. So once activated, B cells become plasma cells. There's also memory B cells. Memory B cells, these are the veteran workers that remember past infections and allow for rapid, stronger immune responses upon re-exposure. B cells can take different paths based on how they are activated. T cell independent activation or a weak response, some antigens like bacterial polysaccharides, examples are in Streptococcus pneumoniae, can directly cross-link surface IgM or IgD without T cell help. The key features, they have a fast response, but mainly IgM antibodies are produced. No memory cells are generated, so there's no long-term immunity, and seen in the response to capsulated bacterial. This is why vaccines for encapsulated bacteria, like pneumococcus, need polysaccharide protein conjugation to recruit T cell help and create a stronger immune response. Then there's T cell dependent activation, which is a stronger response. For most antigens, B cells require CD4 positive T cell help to fully activate. This is the gold standard for long-lasting immunity. The key steps for activation. B cells present antigens via MHC class II to CD4 positive T cells. Then what happens is a CD40 on B cell binds with the CD40 ligand on T cells. This is the second activation signal, so they need two signals to activate. T cells then secrete cytokines like IL-4, IL-5, IL-6, or IL-10. These are examples that lead to class switching. So the antibodies can switch between different classes like IgM to IgG or to IgA or to IgE. There's also plasma cell differentiation, which are the antibody factories. Active B cells switch gears to produce the best antibody class for the job. So B cells can switch into different types depending on the signals they receive from T helper cells. Think T helper cells help your B cells know what to switch to be able to fight the specific antigens. So an example that I created for you guys, so the different types of immunoglobulins that are produced by B cells are on the left-hand side. So these are the antibodies that your body produces, and each of these antibodies have different functions. For an example, IgM, this is your first responder. I think of it as the Ig Marine, the Marines that are in high amounts. They are the first responders and also a complement activation. However, what makes IgM different is that they are T independent activation. They do not need T cells. They are in abundance. They are the Marines. The next is the IgG, or the gangster, the Ig gangster. Why? Because IgG can cross the placenta. Why does that matter? Because when a baby is born, they have passive immunity for 6 months due to the IgG passing from the mother into the baby. The T cell signals that are needed to activate IgG include IL-4, IL-6, and interferon gamma. The next antibody is IgA. This is your mucosal defense, like in your tears, saliva, gut, breast milk, etc. If there's a deficiency, there'll be recurrent mucosal infections. The T cell signals they need are TGF-beta and IL-5. IgE is parasite defense, allergies, and mast cell activation. They're involved in anaphylaxis and type 1 hypersensitivity reactions. They require IL-4 and IL-13 to be activated. The IgD is a little bit of a question mark. The way I remember it is IG don't remember. IgD because we don't know what it does. It's not very well understood. It's likely involved in naive B cell activation. So some high yield tips. Remember, class switching occurs at the DNA level by cutting and recombining genes. Hyper-IgM syndrome, for an example, results from a CD40 ligand deficiency. Remember, B cells need 2 signals to activate. The second signal they need is from CD40. If they don't have this second signal, it prevents class switching, so there's only IgM. Remember, B cells need a second signal, and IgM are the Ig Marines. There's also memory B cells, remember, and secondary response. Think of memory B cells as experienced warriors who remember past battles and immediately counterattack. Vaccines work by generating memory B cells. So what happens, we inject an antigen into the body, your B cells create a formula to remember the antigen. So a quick summary, naive B cells are activated by antigen binding or CD4 positive T cell help. If T cell independent, they only secrete IgM or the Ig Marines. This is our weaker response. If T cell dependent, they undergo class switching, hypermutation, and memory formation. Plasma cells are the cells that secrete large amount of antibodies. They are the activated B cells. In the same context, we also have memory B cells, which allows for rapid, stronger response upon re-exposure. The signals that are required is CD40 to CD40 ligand interaction. The class switches from IgM to either IgG, IgA, or IgE, and then they strengthen the memory B cell formation. So this was a high yield, very condensed video. Let's do a quick summary. So B cells are your body's defense system against invaders like bacteria and viruses. They start as naive cells in the bone marrow, but once activated, they transform into plasma cells, which produce antibodies to fight infections. They can also become memory B cells, which remember past infections. So if the same germ attacks again, your immune system responds faster and stronger. B cells are the reason vaccines work. So let's go ahead and do a couple practice questions. Like I will always tell you, read your last sentence first, then read your options, then read the entire question. So I'll go ahead and give you a few minutes and then we'll come back and review the question. So the correct answer was B, Hyper-IgM syndrome. Remember, Hyper-IgM syndrome is caused by a defect in the second signal, which is the CD40 ligand, CD40 on activated T cells. When there's a defect, this prevents proper T cell development. As a result, class switching from IgM to IgG, IgA, or IgE does not occur. Remember, IgM are the Marines. They are the first responders. A common variable immunodeficiency presents later in life with low immunoglobulin levels but normal B cell numbers. Bruton's X-linked A-gamma globulinemia presents with low B cell counts due to a BTK gene mutation preventing B cell maturation. Selective IgA deficiency is characterized by isolated low IgA levels with normal IgG and IgM levels. Severe combined immunodeficiency disorder is a combined B and T cell deficiency, typically presenting with severe infections in infancy. If you don't know any of these specific diseases, I recommend you go check out the rest of our videos. Question number 2, I'll go ahead and give you a couple minutes and then we'll come back and review this question together. So if you chose B, CD40 ligand, that is correct. Remember, it's B cells activation through T cell dependent mechanisms require interaction between CD40 on B cells and CD40 ligand on activated CD4 positive T cells. This interaction triggers class switching and differentiation into plasma and memory cells. Without this signal, B cells remain restricted to IgM production, as seen in hyper-IgM syndrome. CD28 to B7 is involved in T cell activation, not B cell activation. TCR MHC1 refers to a CD8 positive T cell activation. B cells present antigens via MHC2, not MHC1. PD1 PDL1 is a checkpoint inhibitor interaction that downregulates immune responses. IL-2, IL-2R is important for T cell proliferation, not B cell activation. Again, this is a high-yield video. I recommend you go and check out some of our other videos. Question number 3. I'll go ahead and give you a few minutes and then we'll come back and review the question together. So if you chose A, IL-4, that is correct. IL-4, primarily secreted by Th2 cells, is the key cytokine that promotes B cell class switching onto IgG and IgE production. In response to vaccination, T cell dependent B cell activation leads to IgM producing plasma cells switching to IgG production. IL-5 enhances IgA production. C, IL-10 is anti-inflammatory. Interferon gamma promotes Ig2A and Ig3 production. So that concludes our video for today. I know this was a very condensed high-yield video on B cells. If you don't know any of the other disease mechanisms we talked about in our practice questions, I recommend you go and check out our other videos and also the healingpowerofnature.com and also our Patreon. We appreciate your support and look forward to seeing you next time.